I'm Srinand Paruthiyil, and my project this year was the assembly of the histoplasma encapsulated genome with optical mapping. I'm a rising senior at Lowell, and my mentors for this project were Yvonne Lai and Angel Mack. We had collaborators from the SIL Lab, Srinand Paruthiyil and Mark Voorhees, and my PI was Huiyan Kwok. So it's actually my second year at the Kwok Lab. Here's a little picture from last year's symposium. And I was in the cardiovascular research building. And here's a bit of my experience. I started the LSRP program in sophomore year, where I learned how to present and read science just to get introduced to the world. I then interned in the Kwok Lab doing mainly computer work, and I've come back this year, and I did both computer and wet lab work, but today I'll be talking mainly about my computer work. So what is histoplasma? It's a pathogenic fungus that's found in bird and bat droppings, and it enters the human body through inhalation. It primarily affects the lungs, where it's ingested by macrophages and can be spread to the rest of the body. It can be go to die, it's pretty terrible, and it's dangerous to those with compromised immune systems mainly, such as those with HIV/AIDS. And with over 1 million people having that disease, it's a pretty major issue. So a fully annotated genome would give us a better understanding of the disease and could lead to some potential treatment. So a little analogy for our project is that optical mapping works as a jigsaw puzzle's picture, and it aligns together the jigsaw puzzle pieces which represent the sequencing contig. Here's a little better explanation, where red represents an actual optical mapping contig and blue represents the sequencing contig. So optical mapping visualizes every single location of a specific sequence of DNA. In this case, we use the enzyme BBVC1, which nicked at every location of GGAGTCG. And you can see that this location of GGAGTCG aligns with this location, this one with this one. So our methods are hold on one second. So that's really pretty. We have DNA going through nanochannels that visualizes every location of the BBVC1 nicking site, previously GGAGTCG. And once you have that, you eventually get an optical map which shows you the location of every single one of those nicking sites. So each line here in this barcode sort of looking thing represents a nicking site. And this pattern of nicking sites matches with the pattern shown by the sequencing contigs. So you know that that's the order of the sequencing contigs. So our results, we have de novo assembly attempt ASM5, which has covered 70% of the sequencing contigs. Now, my next step, which this is what I mainly did this year, was aligning the genes found through microarray to our assembly. So we want to find the locations of the gene in the genome. So we first had to prepare ASM5 a little bit, where you have gaps between the sequencing contigs, and they need to be filled with N nucleotides, which represent any nucleotide can be here. It's just we know that it's this length of a gap. And so you fill those in with N nucleotides using scripts. Then the next step of the preparation for annotation is you have to take out inconsistencies. So you can see in this green sequencing contig, there is a continuous line of nick sites, but here there's a gap, and that's an inconsistency. So we take out that whole optical map so it doesn't cause any problems in our continuous DNA file. So our results, we have an assembly that's providing gene cluster information as opposed to the individual gene locations, which can already be shown through microarray. So you can see that gene W goes here, gene X here, see. And then you have gaps represented by N's. So in conclusion, we have an accurate assembly of the majority of the genome, and we have further proof that the use of nicking sites as a genome motif is very effective in de novo assembly. And optical mapping can provide an overview of the distribution of genes in a genome. Future directions to maybe produce a better assembly is to use a different nicking enzyme to cover contigs without any nicking sites. So you'll often have regions that have very few or no nicking sites, so you can't align any sequencing contigs there. So using a different nicking enzyme could cover those regions. So I'd like to thank Richard Shapira, Julia Yee, Lauren Meyer, all of those in the Lowell Science Research Program for giving me this awesome opportunity to work here. I'd also like to thank Huiyan Kwok, Angel Mack, Yvonne Lai, Catherine Chu, Annie Poon, Chinlin, and every other member of the Kwok Lab for mentoring me and dealing with all of my stupid crap throughout this whole process. And really, this has been awesome. I came up here last year and I said, in like the most nerdy voice ever, bioinformatics is the coolest thing ever. And you know, now I think it's not just that, because you can read a book and I can learn bioinformatics, but coming here every day and really immersing yourself in the whole experience. And that's what's really made this experience special. So thank you so much.